Hi guys, Professor Ruggieri here for the third lab of this week, which is going to be the Lysozyme Lab. So anything that ends in zyme tends to be an enzyme, right? Some sort of protein that catalyzes a chemical reaction. Lysozyme is a naturally occurring enzyme. We have it in our tears, our saliva, and it helps to actually break down the peptidoglycan in bacterial cell walls. So it's antimicrobial. So the overall purpose of today's lab is to basically examine the action of naturally occurring lysozyme on Serratia marcescens. So we're going to use our broth culture that we used for aseptic technique and for the isolation and spread plate technique. So for this lab, you're going to need one of those plastic clear cups from your kit. I labeled it egg white because that's what it's going in, right? And you'll also need an egg. You'll need your alcohol solution, and we're going to do this now so it leaves a little time to air dry. What it says to do, and I'm actually going to just put a little alcohol right inside the cup, okay? And I'm going to roll it along the edges so that it contacts, you know, it's 100% contact without having the cotton paper towel in there just yet. Why am I doing this? Because I need to sterilize the inside of this cup. So I'm going to switch it around. I'm going to pour it down the sink. And I'm actually going to leave it inverted while I go over a few other things, and then we'll wipe it dry with a clean paper towel. This surface is clean, but I'll pull another one off the roll. Okay? I'm going to take out my egg, and actually one thing to do beforehand that I recommend is I'm going to put a little bit of the isopropyl alcohol on my gloves and just rub back and forth, and then just kind of blot dry on the paper towel so that I'm not touching the eggs with any sort of yuckiness on my gloves. Okay, plus, if these are presumably eggs that your family eats, you want to be careful. So I'm going to take out an egg, put it right here, and I have my plate. Okay? For this, unlike the isolation and spread plate technique, you're not going to dilute into various thirds or quadrants. Instead, you're not going to use the loop. You're going to use one of your sterile cotton swabs. You're going to dip it in the broth of Serratia marcescens and try to cover, okay, by rotating it 90 degrees each time, just cover the entire plate with bacteria. You're also going to need, and I'll show you the bag that it's in case you're kind of lost. You should have received 2 bags that look like this, 2 green bags, okay? And this one said this small items bag contains Kirby Bauer disc bag, Lysozyme disc bag. This is the Lysozyme disc bag. It says blank sterile discs PK/2. All right, so you're going to take that out. These are just paper discs, and you should also have metal forceps in your kit. They do call for dipping your forceps in alcohol at one point, and even though alcohol is a sterilizing solution, especially if this is alcohol that you are then going to use on your family or in your house for any reason, don't dip your forceps directly in here. I just grabbed a red Solo cup that we had and just labeled it alcohol. Sorry for the sirens, okay? And then I'm going to dip my forceps in here. And I'm actually going to give them a dip right now just to get them sterilized, okay? And let them sit right here. So I think we have everything. The only thing I want to do before we get started to save myself some time is label my nutrient agar plate. So this is going to be S-MAR. And we already have an S-MAR plate over here for the aseptic technique. So I actually might just write on this one aseptic, but chances are you've already done this one. So on here, let's just put lyso or lysozyme. You can just put lyso if you want. My initials KR, the date 5/31, and the fact that this is nutrient agar NA. Okay. So let's prepare our egg white. Now, why are we using egg white? Okay, we're using egg white because, and this was just a clean enough paper towel, I think, just going to dry it out a little bit. Egg white has naturally occurring lysozyme enzyme in it. So we basically want to test the ability of the lysozyme that comes from the egg white to ward off, to create like this halo or zone of inhibition where it kills off the gram-negative Serratia marcescens bacteria. So I'm going to crack this, and I'm doing it right in front of you because it's probably not going to go extremely smoothly. Okay, you don't want to get any of the yolk, And remember, you don't need much of the egg white. So I'm just gonna leave that. Oh boy, okay. I think I got a little bit of yolk, that's okay. And remember, this has salmonella bacteria, so you wanna clean this up thoroughly as soon as this is done. But to keep it real, I'm gonna let this go. Actually, follow me while I just wipe my hands off a little bit. Okay, and grab a paper towel here so this doesn't spread everywhere. And I will sterilize this down right after. Okay, I'm gonna open up my package of the sterile cotton swabs, and they come 2 swabs per package, but I don't need 2 right now, so only open it a little bit. Pull out the one that you need, and then reseal it. Okay? Now, I have my egg yolk ready. Okay, this is, egg white ready. And then I have my mixed culture, my broth culture of Serratia marcescens. So I'm gonna tighten the cap a little bit. I'm gonna invert it to mix it up. Okay, I'm gonna flame the lid if my flame was going. And then I'm gonna dip my sterile cotton swab in there and let it soak for a second or 2. Take this back out, flame the lid, put this back over here, and then take my agar plate and streak it literally from supposed, wall to wall. I know it's round, but from edge to edge completely, as if you wanted to put frosting all over the surface of a cake without leaving any exposed cake. Okay, and you don't want to apply much pressure because you're going to end up gouging the agar. Okay, and you can actually see the streaks when you're doing it in person. So you want to see if you can overlap as much as possible. I'm gonna dip this in here, okay, and this is the first time I'm gonna leave this open for a second. Now my forceps have been sterilized already once, so I'm gonna reach into the bag. Okay, the inside of the bag is presumably sterile. I'm gonna grab one of these discs, actually, because you might run into the same issue. I'm gonna put it right back in here for a minute, cover this back up, and I'm actually gonna draw 2 circles on the bottom where I wanna place my discs and label which one is which, because chances are I'll forget. So this is gonna be C for control. That's the disc that gets soaked in nothing, it's dry. And this is gonna be egg. Okay, it's important that we do that, and showing you what happens, mistakes in real time. So flip it back over, okay? Maybe dip them in the alcohol one more time. And what they actually want you to do, and I apologize, maybe I can get this to work. If not, I'll just use the lighter, but don't copy this, it's not safe. Okay. I'm gonna dip this in alcohol. Whoops. Light the lighter and flame the ends to sterilize them. Okay. They will char a little bit, that's okay. Let them cool off for a minute or 2. Okay. Gonna go in here. Gonna grab one of these discs. I'm gonna dip it in my egg yolk solution. Egg yolk, I keep saying egg yolk, egg white solution. Okay? And then I'm gonna take it out and I'm gonna place it right on where I had written egg. Easier said than done. Oops. I am actually, let's see. There we go. Push it on there and smush it down. And I really should not have dragged it. I should have just left it where it was, but it's on there. I'm gonna put my forceps in the alcohol again to sterilize them. Okay, flame them. You can actually see that they're on fire because it's burning off all the alcohol. Okay, kind of go like that to get rid of, sometimes it'll just resurge again. And what's kind of weird about this, I'll be honest, is in a traditional real setup, you wouldn't have a dry disc and then one that was wet with egg white. You would probably dip this one in sterile water, but we're gonna follow the protocol. So you're just gonna pick up the dry disc, put it on the control spot, push it down, okay? Leave it, and then remember, well, in this case, we're not gonna invert your plate, regardless of what it says in the manual, because I don't want these discs to slip. So this is the only one, but you can see some condensation. Maybe on mine, I will try to flip it. Let's see what happens. They seem to be on there pretty good. So yeah, you can go ahead and flip your plate. I'm just worried about too much condensation on the lid, especially with it being humid. So now this is done, and basically we're gonna look to observe if we have this gap in the growth of the Serratia marcescens in the vicinity of the discs. Now, just before we finish this up, you're gonna do your traditional cleanup, right? You're gonna get rid of this. It is egg yolk, so you wanna put this down the drain or in the toilet and flush it, and then bleach everything, sterilize everything here. These cultures, your broth culture of your Micrococcus luteus and your broth culture of your Serratia marcescens, if you just leave the caps a little bit loose, teeny bit, but secure, you can put these in the refrigerator, and you can use these in later experiments. The tube of the mixed M. luteus and S. marcescens, you likely don't need to keep. You can sterilize. I have my sterilization container here. I'm not actively using it with the griddle there. But you can probably get rid of this. These, the broth culture and the nutrient agar, or the tryptic soy agar slant from the aseptic technique, have to be kept at room temperature. Okay? So I will see you for the next lab experiment.